Okay, so we're going to start Chapter 8, Section 1. As you can see, there's several sections in this chapter. We'll cover most of them in notes, but 8.2 we're actually going to cover in a chart format because we're looking at scientists and what they did. So we'll look at the video tomorrow in class, but to start off, this first section is just dealing with an overview of the process of photosynthesis, and clearly we know that photosynthesis is responsible for all of the energy that we utilize on Earth in the form of food. So our producers take that solar energy, that sunlight, and convert it into a form of chemical energy that everything uses. This is our formula for photosynthesis, so it looks real familiar, or at least it should. If you just reverse the arrow, that would be the formula or the equation for cell respiration. So instead we have our reactants being carbon dioxide and water, and our products are glucose, water, and oxygen. So when we talk about photosynthesis, we always have talked about oxygenic photosynthesis. We've just never called it that. But oxygenic photosynthesis is carried out by the majority of autotrophs. So cyanobacteria, which we find in our aquatic ecosystems, 7 of the groups of algae produce oxygen through photosynthesis, and then all of our land plants that contain chloroplasts also produce oxygen through photosynthesis. So when we're talking about that process, the process of photosynthesis, there are really 2 main reactions: the light-dependent reactions, otherwise shortened up to the light reactions, and then the light-independent reactions. Some people call it the Calvin cycle, or some books call it the Calvin cycle. Some books call it the carbon fixation reactions. I usually just call it the Calvin cycle. So the light-dependent reactions clearly are named because they require sunlight. And as autotrophs and photosynthetic organisms, they have the ability to capture that light, which nothing else does, right? We all can feel the energy of the sun, but we don't capture it and utilize it to produce chemical energy like plants can. The purpose of the light-dependent reactions then is to capture that energy and to make the products for the next stage of reactions called the Calvin cycle. So the light-dependent reactions do not make sugar. However, they make the energetic products that we need so that the Calvin cycle can make sugar. And so our second phase of the process, carbon fixation reactions, or light-independent reactions, or the Calvin cycle. Used to call these the dark reactions, but that name was a bit misleading. And I'm sure there was a group of scientists sitting around arguing about this somewhere, but we don't call it the dark reactions anymore because that name indicated that they took place in the dark if the name of the light reactions indicate that they take place or need light. So the dark reactions do not require the absence of light. They just can take place without light. So that's why we call them the light-independent reactions. And what they do is they utilize those energy molecules from the light reactions, and they make glucose as well as waste water, but they utilize the energy from the light-dependent reactions, and they make sugar. Now, this process can't happen without that specific organelle called the chloroplast that all autotrophs have. Specifically, 2 areas within the chloroplast, the thylakoid membrane and then the stroma. So as you look at a chloroplast, there are specific structures. So first off, the thylakoid membrane, that is the internal membrane, the little individual coins or individual sacs that make up the grana. Then the grana is a stack of those thylakoid membranes organized, right? So the light-dependent reactions take place all along the thylakoid membrane because it contains chlorophyll clustered with other accessory pigments into what we call photosystems. Photosystems, I think of them as a cluster of solar panels absorbing sunlight. So there are photosystems, 2 of them, that are absorbing the energy of the sun and transferring that energy to electron carriers. Now, so you have the thylakoid membrane, which is the individual. You have a stack of those thylakoids, which is the grana. And then all of those grana are interconnected by membranes called stroma lamella. And so that's just how they are connected and how they can transfer and share, how they are interconnected, and they can transfer and share, you know, reactants if necessary. The stroma then is that semi-liquid fluid that surrounds the thylakoid membrane, and it's in the stroma where the Calvin cycle takes place. So in this picture here you see a cross-section of a leaf, right? The epidermis, there's an upper and a lower epidermis, and then the middle section is called the mesophyll, and the mesophyll contains cells that have chloroplasts. So each individual cell there, you can see, has several chloroplasts within them, and the major photosynthetic layer is the palisade mesophyll with the spongy mesophyll right under it, which does do photosynthesis, but it's also very important for gas exchange, as well as the stoma or the stomata surrounded by guard cells there. Then if you take one of those cells and blow it up, right, you can see where the chloroplast is in there, or how many of the chloroplasts there are. And then if you blow up a chloroplast, there you see the thylakoid disc and the grana, which is the stack of thylakoids, the stroma lamella, which is connecting all the grana, and then the stroma, which is the fluid. And of course, we still have two membranes on this organelle, an inner and an outer membrane. When you're looking at this image is showing us kind of how the two reactions are related. So you have the light-dependent reactions that take place on the thylakoid membrane, which is that green bilayer here on the top. The big blue structures with the chlorophyll in it are the photosystems collecting the sunlight. The products of those of the light-dependent reactions then are ATP and NADPH, which you see in the middle of that picture. Those products go to the Calvin cycle and are used to energize and reduce carbon dioxide into, well, for the most part, glucose. We say organic molecules, but for the most part we mean sugar.